Neuro and muscle physiology, membrane transport. The plasma cell membrane. Every cell in our body is surrounded by a membrane. This membrane is composed almost entirely of transport proteins and phospholipids. The primary purpose of a cell membrane is to separate the intracellular compartment from the extracellular environment and to control passage of some materials and restrict the passages of others. Types of membrane junctions. Individual cells connect together to form tissues. These connections are referred to as junctions. Tight junctions are just like they sound. Tight junctions hold the individual cells so close to each other that they prevent molecules from passing through the intracellular space. One example where you would find tight junctions in the bodies is the intestines. Desmosomes, also known as anchoring junctions because they anchor one cell to another to form a tissue. They do a good job of holding the tissue together while allowing the tissue to stretch without tearing. A good example of desmosome junctions is in the skin. They allow the skin to stretch over a joint with movement with holding the tissues together. The final junction is a gap junction. Gap junctions allow communication between cells, specifically allowing ions to flow between them. Tissues that have gap junctions for communication will typically have another type of junction holding the tissues together. For example, cardiac muscle have both desmosomes and gap junctions in the intercalated disc to hold it together and allow ions to pass. Cell physiology. Most body cells can metabolize nutrients, digest foods, dispose of waste, repair, grow, reproduce, and move and respond to a stimulus. In order for a cell to carry out the processes noted above, substances must be transported into and out of the cell and within the cell. Body fluid compartments. The body has two main categories of fluid in itself, extracellular fluid and intracellular fluid, but it has three fluid compartments. The extracellular compartment, which I will always refer to as blood when lecturing, the interstitial fluid compartment, which is the fluid that immediately surrounds the outer portion of a cell's membrane, and the intracellular fluid compartment, which contains the cytosol inside a cell. These compartments are separated by structures such as the tunica layers that make up a blood vessel and the cell membrane that surrounds a cell. Familiarizing yourself with organization of these compartments is necessary in understanding the movement of molecules such as nutrients, water, and waste products that occurs between these compartments. The rule of movement of molecules in the body is that molecules tend to move on their own kinetically from an area of high concentration to an area of low concentration. This is referred to as down their concentration gradient. Selective permeability. Cell membranes are selectively permeable, meaning that the cell itself can determine what passes through their membrane. Some molecules are permitted to pass through the membrane while others are not. There are two types of membrane transport: a passive process that does not require any energy at all, and an active process which does require energy in the form of ATP. Passive process of membrane transport. The two types of passive membrane transport are diffusion and filtration. Passive diffusion is when molecules, or I could say solutes, move passively across the cell membrane from an area where they have a high concentration to an area where their concentration is low. Filtration is the process by which water and solutes are forced through a membrane by hydrostatic pressure. Types of diffusion. There are three types of diffusion: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is exactly how it sounds. Molecules or solutes simply move kinetically without assistance through the phospholipid portion of a cell's membrane. Facilitated diffusion, on the other hand, requires assistance of transmembrane proteins interspersed through the cell's plasma membrane. There are two types of transmembrane proteins that assist with facilitated diffusion, and they are carrier-mediated proteins and channel-mediated proteins. Osmosis is unassisted passive movement of water across a semi-permeable membrane. We'll cover more about osmosis a little later on in this presentation. Simple diffusion. Simple diffusion is pretty much limited to lipid-soluble solids and really tiny molecules such as oxygen and carbon dioxide. Here again, I'm reminding you that the natural movement of molecules is always away from high area of concentration to low area concentration until equilibrium is reached across the membrane. Facilitated diffusion. Facilitated diffusion helps to move large lipid insoluble substances such as glucose and amino acids and ions into the cell through a transport protein in the cell's membrane. As with simple diffusion, these solids move through a transport protein from an area of high concentration to an area of low concentration without requiring any energy. Facilitated diffusion is regulated by the activity and/or number of individual transmembrane proteins located within the cell's membrane. The cell's own DNA determines the number and type of transmembrane proteins it produces. The activity of these transmembrane proteins is determined by specificity. Each protein is designed to allow transport for specific molecules such as amino acids or potassium ions, for example. Therefore, a carrier-mediated transmembrane protein designed to only transport amino acids cannot transport glucose. And a channel protein designed to only allow the passage of potassium ions cannot allow calcium ions to pass through. Saturation also plays a role, particularly with carrier-mediated transmembrane proteins, as they can only transport so much within a given time frame. And the stimulation of channel transmembrane proteins by a neurotransmitter has a significant effect on that protein's activity. Carrier-mediated facilitated diffusion. If you look at the diagram, you will see that the process of carrier-mediated facilitated diffusion involves a molecule attaching to itself to a specific binding site on the carrier-mediated transmembrane protein. Then the protein changes shape and essentially dumps the product into the cell. Channel-mediated diffusion is the most pertinent type of transport we will cover in this course, as this is the type of transport mechanism and transport protein used in the stimulation of a neuron and a muscle cell. Channel-mediated facilitated diffusion involves the movement of ions across the plasma membrane through a protein channel into a cell. There are two types of protein channels. Leakage channels, which are always open, allowing ions to move at will down their concentration gradient, and gated channels, which are controlled by a chemical stimulus such as a neurotransmitter or electrical signals such as depolarization of the cell's membrane. Ligand-gated protein channels, also known as ionotrophic receptors, are chemically controlled channel proteins. Ligand means the bonding of a chemical. Ligand-gated channel proteins are also classified as receptors. These proteins are activated by chemicals, and for our purposes, that chemical will be a neurotransmitter. When at rest, ligand-gated protein channels are closed. However, when a neurotransmitter chemical messenger docks with a specific binding site on a protein channel, it stimulates the channel to open, thus allowing ions to flow in or out of the cell depending on the concentration gradient of the ions involved. Voltage-gated protein channels are activated by changes in the membrane potential, specifically when the membrane becomes depolarized. Depolarization of a membrane occurs when the inside of the membrane becomes more positively charged than the outside of the membrane. Aquaporins. Water is a polar molecule, therefore it cannot easily pass through the phospholipid portion of a cell membrane. But it can pass through the phospholipids if the osmotic pressure is strong enough. However, when a body wants to move water around between compartments without depending on osmotic pressure, it can do it through these specialized channel proteins called aquaporins. Aquaporins are pretty much limited to the kidneys and require stimulation by the antidiuretic hormone to be activated and open. Diffusion of water through a cell membrane, either through the phospholipid layer or through aquaporins, is called osmosis. Osmosis is the movement of water through a selectively permeable membrane. Water follows solids, or stuff, and therefore will move via osmosis in the direction where the solids are more concentrated. Solids on the more concentrated side of the membrane will literally draw the water through the membrane until the solids on both sides of the membrane are equally separated by the water molecules. Movement of water in the body. When osmosis occurs in the body, water either enters or exits the cell. The osmosis of water can change the size and shape of a cell. Changing the size and shape of a cell affects the tonicity of the cell. Isotonic conditions inside the body. The prefix iso means same. So an isotonic condition is where the concentration of solutes to water is the same in the extracellular fluid of blood vessels, or blood, as the interstitial fluid surrounding the cell and the cytosol inside the cell, meaning all three compartments have the same ratio of solutes to water. Under normal, healthy conditions with normal body fluid levels, all three compartments should have an equal distribution of solutes to water. In isotonic conditions, cells retain their normal size and shape as water will just flow in and out of the cells in equal amounts. Hypertonic conditions occur when the concentration of solutes is greater in the extracellular interstitial fluid compartments than the concentration of solutes in the cytosol. This situation occurs with dehydration or hyperglycemia, hypernatremia, high cholesterol, or severe vomiting and diarrhea. Really, any condition that will cause the blood to have more stuff than water can cause hypertonic conditions. The high concentration of solids in the blood will have an osmotic pull on the water in the cells in effort to equalize the fluid compartments. This movement out of the cells will cause cells to shrink. In this case, cells can regain their size and shape as soon as adequate amounts of fluid enter the body, either through the mouth or through the IV. Hypotonic conditions occur when the concentration of solutes is greater in the cytosol than in blood. Water will be drawn into the cell until the distribution of solids is equal between all three compartments. This type of situation can be lethal, especially in the brain. Cells are tiny and the vascular system is huge. If hypertonic conditions are severe, as in the case of overhydration, the osmosis of water into a cell can cause it to rupture. Severe overhydration can cause death if brain cells begin to rupture. Treatment for this type of condition would be administering sodium through an IV. Active process of membrane transport. Unlike passive transport, active transport requires energy in the form of ATP. However, like facilitated passive diffusion, active transport uses specific carrier proteins to get the job done. These carrier protein pump solutes across the cell membrane against their concentration gradient. That's the key thing. Active transport is against the concentration gradient. Anytime you hear or read the word pump when describing the transport of substances, think active transport. Because the transmembrane proteins involved with active transport actually pump the solids, they are often referred to as solute pumps. Now, many of the solids that are pumped across the membrane are ions with either negative or positive charge. So active transport also pumps ions against their electrical gradient as well as their concentration gradient. I basically already said all of this in the previous slide, sorry. Active transport process. Solutes bind to specific receptors on carrier proteins. Energy from ATP causes that carrier protein to change shape. Solutes are pumped across the membrane against their concentration gradient. Very similar to facilitated diffusion. The only difference is facilitated diffusion doesn't require energy and it goes down the concentration gradient, whereas in active process, it's going against the concentration gradient. There are several primary active transport pumps in the body that are necessary to sustain normal functions, such as the calcium pump, hydrogen pump, chloride pump, and sodium-potassium pump. In this course, we're going to focus more on the calcium and hydrogen pump and sodium-potassium pump, not so much the chloride pump. But they all do exist, and they are all important in helping cell membranes maintain their concentration gradient and electrical gradient across the membrane. Sodium and potassium. Principal cations in the body are sodium and potassium. Inside all cells, the concentration of sodium is low and potassium is high. On the outside of cells, it's the opposite. The concentration of sodium is high and potassium is low. This concentration gradient of sodium to potassium inside and out of the cell is maintained by the sodium-potassium pump. The sodium-potassium pump continuously pumps 3 sodium ions out of the cell while simultaneously pumping 2 potassium ions into the cell against their concentration gradients and/or electrical gradient. So where's all that sodium potassium coming from? Remember the leaky channels that we talked about with facilitated diffusion of ions through the plasma membrane? Well, cell membranes are full of these leaky channels. Sodium-potassium leaky channels. Cell membranes are filled with these leaky protein channels that are specific for either sodium, potassium, and these leakage channels are always open, and that allows ions to flow down their concentration gradient continuously. Concentration and electrochemical gradients. Sodium and potassium will continuously leak through the cell, through the leaky channels in the plasma membrane down their concentration gradient until concentration equilibrium is reached or electrochemical gradient is established. The normal function of the sodium-potassium pump will prevent the ions from ever reaching chemical equilibrium, which is critical for maintaining membrane potential. And we'll cover membrane potentials later on in this course. Did you notice on the previous slide that there are twice as many leaky channels as leaky potassium channels as there are sodium channels? So what's preventing all the potassium from leaking out of the cell faster than the sodium-potassium pump can pump it back in? What is keeping potassium in is its affinity to bind with intracellular free amino acids, meaning only free potassium will leak out. This binding of potassium to amino acids is responsible for establishing the electrochemical gradient across the cell membrane. Vesicular transport is an active transport process that requires energy. Vesicles merge with cell membranes, so substances do not actually cross the membrane. Vesicles transport large molecules such as proteins, neurotransmitters, hormones, mucus, pathogens, etc., etc. There are 2 types of vesicular transport: exocytosis, which moves substances out of the cell, and endocytosis, which moves substances into the cell. Exocytosis is the process of active transport that begins with a sudden increase in intracellular calcium. This is a very involved process that we will discuss when we cover synapses in this course. This increase in intracellular calcium will stimulate the vesicles to fuse with the cell membrane and release its contents outside of the cell. Some of the substance released by exocytosis are hormones, neurotransmitters, mucus, and waste products. And we'll be mostly focusing on neurotransmitters. V snares and T snares. The process of vesicular exocytosis is extremely involved, and as I mentioned before, we'll cover this process in detail when we get to the topic of synapses. For now, all I want you to know is that vesicles have these specialized proteins sticking out of them called V snares, and the V stands for vesicle. And the cell membrane has these specialized transmembrane proteins protruding into the cell called T snares, and the T stands for transmembrane. These snares play an intricate and vital role in the binding of a vesicle to a cell membrane. G-protein linked receptors are a specialized 7-membrane transmembrane protein that has a G protein linked to it. This specialized receptor is utilized by many neurotransmitters and hormones that we will cover in this course.